Hey everybody, we are going to continue on with our chapter from gene to protein, and we are going to talk today about transcription in some more detail. So this is the process of making RNA from a DNA template, and this should look pretty similar. So we've got our very basic steps of transcription, and what you should notice is that they look pretty similar to the steps that we followed with replication. A big difference though is because we're making RNA, so we have RNA polymerase instead of DNA polymerase. So step one is going to be the binding of RNA polymerase. Then we have initiation, elongation, and termination. And so RNA polymerase is going to be an enzyme that's used for building RNA from RNA nucleotides. Again, we talked about replication and how you build DNA, and that building DNA polymerase 3 was used to build DNA from DNA nucleotides, so RNA polymerase does the same thing for RNA. So now binding requires that the enzyme find the right place on the DNA to attach and start transcription. This is going to be one of the big differences. With DNA replication, we had origins of replication all throughout that DNA molecule because the entire molecule needed to be copied. When you are looking at transcription and creating RNA, you're creating RNA for certain genes or certain locations on that DNA molecule, and so you need to find the right spot on the DNA in order to start replication in the correct area. So binding is a complicated process. It uses promoter regions on the DNA. These are going to be regions upstream for the information from the protein, and it's going to be like a signal saying like, hey, bind here, this is where the gene is. And it requires proteins that are called transcription factors. So here we have our DNA segment in the light blue over here, and then you've got the promoter region in green, and then the dark blue is going to be the transcription unit or the DNA of the gene. This is exactly what we want to copy. Then you've got a terminator sequence, and then termination will actually happen right here, and then the DNA molecule would continue. So how does this work? How does the RNA know where to start being copied? And so for that we have what's called a TATA box. And so TATA, so thymine, adenine, thymine, adenine. And this is going to be located 25 nucleotides upstream for the initiation site. And this is the recognition site for transcription factors to bind to the DNA. So if we go back really quickly, we've got our promoter. We've got what we want copied, the actual DNA of the gene. We need a way to sort of flag that RNA polymerase and say like, hey, this is the DNA you need to copy to get the RNA. And so that happens with the TATA box. And so here you can see we kind of zoomed in a little bit on the promoter. And so the TATA box is at the beginning of the promoter. So transcription factors are proteins that bind to DNA before RNA polymerase. They recognize the TATA box, they attach, and then they flag the spot for the RNA polymerase to get started. So again, here we are showing this transcription factor. And right now it's sort of highlighting where that TATA box is. And that transcription factor will then signal to the RNA polymerase. So the complete assembly of transcription factors and RNA polymerase bound to the promoter area of the DNA, that's what we call the transcription initiation complex. So you need to have the transcription factors, and you need to have the RNA polymerase, and all that is going to be at the promoter area of the DNA to be transcribed. So what that looks like is right here. So this is our transcription initiation complex. So you can see you've got your RNA polymerase 2, and then you've got your transcription factors, which are other proteins that are binding to this site. And so again, you've got the TATA box, so the promoter region is a slightly darker green. Then you've got the rest of the promoter, and then you've got your DNA that needs to be copied, the DNA of the gene. And so one thing I want you to notice is we're copying this bottom strand of DNA. And you can see that we've already started to make the RNA transcript back here. So if you are copying important genetic information from a gene, you don't want to start too late. So if the RNA polymerase was like starting right at the very start of the dark blue, it might miss something. And so you can see that the start point is actually still within that promoter region, so it does start copying before it actually hits the DNA of the gene. Next we have initiation, and so initiation is going to be the unwinding of the DNA to start RNA synthesis, and this requires what we call initiation factors. So we are looking down here, and we already have our transcription started, right? So we've got our slight RNA transcript, and the DNA has been unwound, and so this is initiation right there. With elongation, the RNA polymerase is going to untwist the DNA one turn at a time. When you untwist it one turn at a time, you're exposing 10 DNA bases for base pairing with every untwist of that DNA molecule. And so for every one turn, you're able to copy 10 DNA bases for pairing with RNA nucleotides. And that is what we're looking at here. So you can see the RNA polymerase is untwisting one section of DNA at a time. As that is happening, you have your RNA nucleotides that are being added to base pair with the template strand of the DNA. What I want you guys to notice too is that this is working in the same direction. So it is reading this template strand from 3', you can see, and then to the 5' direction. So elongation, the enzyme is going to be creating the RNA transcript in the 5' to 3' direction. So again, I know this, we got caught up with this with replication. On the parent strand, you can see the enzyme is moving in this direction, so it's starting at the 3' area and moving in the 5' direction, and then the RNA that's being made is being made in the 5' to 3' end. All right, and so this happens pretty quickly. This is going to happen, about 60 nucleotides per second are going to be added to this growing RNA transcript. All right, so now we've kind of got the three parts together now. So we've got just sort of showing us where the RNA polymerase is on the promoter region, then initiation, so you're going to start to unwind the DNA, you're going to start the RNA transcript along that initiation site in the promoter, and now you've got a pretty long RNA transcript through elongation as the RNA polymerase moves down towards the far end of the gene. So just a quick comment, each gene can be read by sequential RNA polymerases, and so what that means is you can have one RNA polymerase going through this and copying one RNA transcript, but then you can have another RNA polymerase doing the same thing right after it. And so what that means is you're going to get several copies of that protein that can be made because you're copying the gene multiple times. Alright, so now what happens when you get to the end of the gene? You need to be able to terminate that RNA sequence. So the DNA tells the DNA tells the RNA polymerase to stop. Okay, and so then the RNA polymerase is going to detach from the DNA and it's going to close up the helix. So here we have termination. You can see that the RNA polymerase has left and you have your completed RNA transcript. Now this final product we actually call pre-mRNA, so it's pre-messenger RNA, and we call it pre-messenger RNA because it's not quite done yet. So this is a very raw final product, and it needs to be further processed before that mRNA can actually take its message outside of the nucleus into the cytoplasm for it to be translated into a polypeptide sequence. All right, so this is pre-mRNA and it will be processed into mRNA. Okay, and that's what we're going to focus on next time. So your lecture question for today is, I'd like you guys to compare and contrast DNA replication to transcription. So what are some things that seemed very similar between the two processes? Where were the differences? And I want you guys to shoot me a message with your response to that question. Thanks, guys.